In previous videos, I talked about the chemical nature of DNA and RNA, how they're similar, how they're different, and so forth. In this video, I'd like to wrap things up a little bit by talking about exactly what DNA and RNA do. DNA contains the code, the genetic code for making proteins. Cells do not read the entire DNA code all at once. Small portions of DNA are read, and the technical term for this is transcribed. A gene, which is a unit of genetic information, is transcribed from DNA to RNA. And we can see in this diagram how this works. We have the DNA, which has the weak hydrogen bonds between the two single strands, is able to open up. And an enzyme called RNA polymerase is able to take monomers, which are nucleotides, which we've talked about in previous videos, and use those monomers to assemble a polymer, which will be RNA. Now, the DNA will stay in the cell nucleus. It never leaves. The RNA, however, can leave the nucleus. There are actually pores in the membrane of the nucleus, and the RNA can exit through those pores. And then within the rest of the cell, outside of the nucleus, the RNA is then able to be used as a template for making proteins. And this process is called translation. So again, we can summarize with the diagram just above me. We have the DNA within the nucleus that contains the genetic information. And then little bits of this DNA are opened up in a sense to allow for the transcription of RNA. So we're reading one gene at a time. That information is then transferred. The RNA molecule is actually moved from the nucleus and into the rest of the cell. And then the information encoded in that RNA is then processed in a process called translation to make protein. Okay, well that does it for this video. Thank you very much for watching.